Hello everyone and welcome to Scienticaa. Today we'll discuss about Golgi apparatus. So, Golgi apparatus or Golgi complex is a cytoplasmic organelle which was first seen by George in 1867. But since this organelle has the same refractive index as that of the cytosol, it was not clearly distinguished. So, Camillo Golgi in 1898 became the first scientist to differentiate it and to study it in the barn owl and cat. So he studied the nerve cells of barn owl and cat, and he was able to differentiate this organelle and study it as well. Therefore, the organelle has got its name from the scientist Camillo Golgi. Talking about the occurrence, Golgi apparatus is found in all the eukaryotic cells, but there are some exceptions such as mammalian erythrocytes or RBCs, sieve tube elements in phloem, sperms of bryophytes and pteridophytes. Except for these eukaryotic cells, all other eukaryotic cells have Golgi apparatus. How does the Golgi apparatus develops? It develops either from the division of the pre-existing Golgi apparatus. So this can divide and form the another Golgi apparatus, or it can also develop as an invagination of plasma membrane. So the plasma membrane develops in birds, and this invagination can give rise to Golgi apparatus, or it can evaginate from the nuclear envelope. The nuclear envelope can develop outwards, and this evagination can separate out to form the Golgi apparatus, and also from the endoplasmic reticulum. So endoplasmic reticulum can also form the Golgi apparatus. So these are the four modes in which the Golgi apparatus can be developed. Structure of Golgi apparatus. Golgi apparatus consists of flat, disc-shaped sacs which are called as cisternae. These cisternae are membrane-bound, lined, curved vacuoles which are present in the stacks of 6 to 8. So generally 6 to 8 cisternae are present in Golgi apparatus. However, in some algae, the stacks can be in the number of 10 to 20 cisternae. In some insects, it can even go to 30 cisternae in one Golgi apparatus. Cisternae are arranged parallel to each other. So these are the parallel cisternae, and they are generally arranged concentrically near the nucleus. So this is the nucleus. We can see that this is parallel arrangement near nucleus. Also, it has two phases. The first one is the cis phase. It is also called as the forming phase. It lies proximally. It is convex in shape and is also called as the formative or forming phase. The second phase is the transphase, which is concave and also called as the maturing phase. Now, transphase is distally arranged and it is concave. So this is the transphase, which is arranged distally and it is more concave in shape. Now, this concave maturing phase lies towards the plasma lemma. So when we see this eukaryotic cell, the concave phase is lying towards this plasma lemma. Now, cisternae also encloses a space which is called as lumen, and it is around 10 to 15 nanometers in width. The lumen is seen as wider in the cisternae of the maturing or the transphase. So we can see the lumen over here, which is more wider towards the transphase as compared to this cis phase. Now, in between the two cisternae, there is a space which is called as intercisternal space. So these two adjacent cisternae are going to be separated by this space, which is called as the intercisternal space. Functions of Golgi apparatus. The first important function is modification of materials. We have seen earlier that the vesicles are released from the endoplasmic reticulum, and these vesicles are fusing with the cis phase of the Golgi apparatus. Now these vesicles passes through the intercisternal spaces by budding off from the periphery of each cisterna and passing on to the next cisterna. So they successively pass on from one compartment to another compartment and finally reach the trans phase. From the transphase, they are pinched off. The secretory vesicles are pinched off by the process called as exocytosis, and they are released into the cytoplasm. So, modified proteins or materials are packed inside the vesicles and they are released from the transphase. This is one of the most important functions of Golgi apparatus. Another important function of Golgi apparatus is glycosylation. What is glycosylation? Glycosylation is complexing of lipids and proteins with carbohydrates. And what is formed as a result of glycosylation? Glycolipids and glycoproteins are formed as a result of glycosylation. So, terminal part of glycosylation occurs inside this Golgi apparatus, which results in the formation of glycolipids and glycoproteins, and this is also taking place in Golgi apparatus. Next important function is formation of acrosome in sperms. Now, acrosome is a special structure which is present near the tip of the sperm and it contains the hydrolytic enzymes. And these hydrolytic enzymes are responsible for digesting the protective covering of the egg. So, acrosome is also formed by this Golgi complex. After the formation of acrosome, however, the Golgi complex or the Golgi apparatus degenerates. Another important function of the Golgi apparatus is secretion. That is one of the main functions of this cell organelle. Now, various proteins, lipids, steroids are received by the Golgi apparatus from the endoplasmic reticulum. And we have seen that these proteins, lipids, and other substances are going to be modified, concentrated, and packed inside these Golgi apparatus. And then later on, these pinch off and pass out as the secretory vesicles through the process called as exocytosis. So these vesicles are going to be released out of the Golgi apparatus. Now there are some examples of these secretory products such as the digestive secretions, sweat, tears, saliva, mucus by the goblet cells, some neurosecretions by the nerve cells. Also, we can have sticky slimes and digestive enzymes by the plants, which are insectivores. We can have slime and gums waxes from the plants as well. So all these substances that we have talked about, they are all the secretions from the Golgi apparatus. So these were the major functions of the Golgi apparatus. Thank you. If you have liked the video, please subscribe to the channel, share it with your friends, and if you have any doubt or query, please post in the comment section. Thank you.